has plenty of the pathogen, so a conducive soil, into over time some well-known case studies around the world of soils that have become suppressive to the disease and trying to figure out what's going on with that and the roles that the microbes, ultimately it seems to be coming down to the roles that the microbes are playing. And that in this case, with this graphic, they're showing that if you get this immune response initially developed where you're on the road to suppressiveness, when the pathogen, if it gains entry again, that same pathogen, then the soil is pretty much ready to go and be able to defend that much better against the disease. And so the plant should be able to survive. You also see the pie chart showing that diversity of the microbes tends to go up in a suppressive soil. We looked at this visual too to try to get at the shift from conducive to suppressive. So the last topic in the slide set that I'd like to talk to you about is kind of looking at this idea of pathogen control. It's actually work that my postdoc advisor, Dr. Linda Kinkle, was doing when I was a postdoc there. And she has published some papers on this as well. I'd like to tell you this story that her research is showing on if you want to reduce disease in your crop fields and do it sustainably, we need to know what's going on between the plants and the microbes. The particular microbes that we were studying are called Streptomyces bacteria. You're familiar with them, whether you know it or not, because you've taken antibiotics most likely, and they're the way that most antibiotics are still made today. We grow I don't do it, but they grow the Streptomyces in big vats and get them to produce the antibiotic, and then they purify that and sell it. Very interesting bacteria. It's one that I still work with quite a bit, where we're looking at the idea of right now what you're doing is looking down onto a Petri plate that has these colonies. So that's the Streptomyces colony right there. And those black dots that you can see growing on or showing up on the colony, that's actually like pure antibiotic. So the bacteria is making so much antibiotic that's kind of oozing out of their cells, and you can actually, it kind of crystallizes as it dehydrates. So when you grow Streptomyces in the lab, you'll often see that. They have a very interesting life cycle because they form spores that will land somewhere and germinate and grow. And when they do that, they form hyphal-like growth. So Streptomyces actually means bent fungus, unfortunately, but it's really a bacteria. So they named it Streptomyces before they figured out it was a bacteria, not a fungus, but it does have hyphal growth. It really does look like, under the microscope, it looks like a very skinny, petite fungus with these long hair-like strands, hyphal-like strands that will grow into the agar. And then eventually some of the hyphae stick up off the agar. You get these aerial hyphae, and that's when you start seeing kind of these whitish, kind of granular looking aspect to the colony. And then it usually starts out whitish, and then you'll see a shift into some color, some kind of pigment shows up. That's usually the tip off that they're starting to form spores. And so those aerial hyphae start to fragment into spores, and then eventually they'll break free and do it all over again. So they're really unusual bacteria. They love soil. Every soil system around the world has them. And if you have ever smelled that lovely wet soil smell after a rain, that's probably mostly Streptomyces that are producing a certain volatile. It's called geosmin, and it gives that lovely wet smell, wet soil smell that I love. And so when I grow these bacteria in the lab, it smells wonderful. Most bacteria, if you've ever taken a micro class, do not smell good. So I picked a good one for this. So they're interested in using Streptomyces bacteria because when we were doing work, when I was a postdoc there, they were the group of bacteria that were showing up in that suppressive soil. So when we were talking about that potato scab suppressive soil last time, When we went into that soil system and said, how did that soil become suppressive? We found a huge diversity of Streptomyces bacteria. And when we tested them against the pathogen in Petri plate studies, really good inhibition. And so we were studying those for very long stretches of time. Linda collected hundreds and hundreds of these from that particular field and asking, can we use them as biocontrol agents against different diseases? They're good not only against the potato scab disease, but they also inhibit lots of different fungi that cause diseases. And so the idea here is, okay, how do we support these bacteria so they'll stay in the soil and they'll do their job of helping the plant fight off these pathogens? It's a much more sustainable way of dealing with disease than saturating the soil with all those different fungicides or pesticides. So we want to increase their density. We want to increase how well they can kill off the pathogen. And of course, you ultimately want to see suppression of the disease when you harvest your plants. And so the question is, how do we do this? And it's kind of an interesting story, but the plant certainly is going to play a role in this as well. We know, we've been talking about this for a while, how all the exudates that come from the plant are definitely influencing what microbes are there in the soil close to the roots, how many are there, what ratios, what are they doing. And so when she set up this particular experiment, I wasn't involved in this experiment, but she was looking at how much does the plant really influence what's going on with these bacteria and their ability to kill the pathogen that's trying to attack. And so she was looking at what is the plant species, how many of the same, so looking at diversity, and a little bit of nutrients as well. She was looking at that. What impact does that have? So what she did in this study, they had two C4 grasses. C4 refers to how they do the C4 cycle during photosynthesis. It's not terribly important for this, but she had two C4 grasses and two legumes. And legumes are the plants that are well known to have that relationship with bacteria to do nitrogen fixation. And so the ones that start with L are the legumes, and the other two, the A and the S, that you'll see in these abbreviations are the C4 grasses. Then they had them in plots where they were either a monoculture or 4 species together, 8 and 16 species, and testing the soil microbes and looking at their ability to suppress the pathogen. She did some sequencing also, looking at Fusarium, which is a well-known pathogen, and then the Streptomyces are the biocontrol agents. So when she was collecting the data and analyzing it, the relative abundance of some fungi showing up. So they were assessing, in this case, a lot of these fungi that are listed are not so good. And so some are and some aren't. And so she was looking at fungal composition, what's going on here. And you can see there's clearly a difference depending on if it's a grass species or a legume plant species. So just showing that, yes, you definitely have an impact. The plant can influence who's showing up in the soil. It does matter. So getting a handle on that a little bit more, they were doing more studies with that and then asking, so what does this tell us? How should we think about this? So when you are thinking about the different plants that are growing in a certain plot and all the exudates that they are excreting out into the soil, and if you think about a monoculture of plants versus maybe, the high diversity of plants and think about the differences there, you're going to see you would expect a monoculture of plants would have very similar exudate profiles. All the plants, if it's all corn, for example, you're going to get corn exudate, and you're not going to get a lot of differing nutrients that are going into the soil compared to if you had a plot that had 16 different plants in that plot, different species. Here, she's looking at, do nutrients influence species interactions and selection for inhibition, antagonistic phenotypes. So basically, can they kill the pathogen? This was done in Petri plate studies where they were doing inhibition studies. And we've been talking about niche partitioning idea in previous classes. Here, she's looking at niche overlap of the microbes. And how does that, with the inhibition zone of their ability to inhibit or kill off the pathogen, how does that relate to the niche overlap? If they have a high niche overlap, it's more in this area, okay? The proportion of nutrients that are shared. So they were assessing what kind of carbons sources do these microbes use? Do they all use the same kind of carbon or different carbon? And there's ways to test for that. There's very simple tests that you can do in the lab in order to do this. And you can see a pretty nice and highly significant correlation here where if there's significant niche overlap, you get high inhibition, which means the Streptomyces bacteria, if they share a niche, high niche overlap, that means they're fighting, so to speak, for the same nutrients, which in this case makes them kick into antibiotic production and inhibition as a response to that. That's the hypothesis. Okay. And so this is turned out to be statistically significant, highly significant, anything less than 0.05. So if they are sharing the same nutrients, all vying for the same nutrients, their ability to inhibit goes way up. So this is getting at niche partitioning versus niche overlap. So they were looking into this a little bit in more detail. They collected some soil and they were spiking in either glucose at high and low amounts or lignin. So glucose is a very simple sugar. So that is readily taken up by any living organisms that can use it. They'll love it. Lignin, on the other hand, is very complex polymer, needs a lot of work to get it broken down, persists in the soil longer. So they were looking at two different carbon sources. And then they were asking, how does this influence the phenotype of the Streptomyces? Do they ramp up their inhibition abilities or not? So they were running this experiment, and you can see if there's a lot of glucose. So the blue bars, to my eye, they look blue, or a lot of lignin. You can see in general, that's a good thing in the sense that it is making, so number of standards inhibited. What she means there is that she had, in this case, anywhere from the control all the way up to 5 different pathogens that were being tested against. So they were testing the Streptomyces against 5 different known pathogens and just looking to see, how well. And you can see that, you can see that there's some really nice data here showing that you have the Streptomyces is really kicking into its inhibition if you have better, higher levels of carbohydrate in this case. So looking at what plants are there, what nutrients are available, you give them more nutrients. That, I mean, it kind of makes sense, right? You give more nutrients into the soil, they're going to be able to take those in, use those for their own physiology. They'll be able to reproduce that much more and more frequently. I mean, it makes sense in that regard. So niche overlap among populations will determine the strength of selection. So that was an interesting idea. So then they went back and said, okay, what's going on with the plant? And not only the identity of the plant, its species, but also how many different species are in that plot. What's going to happen if we do this, thinking about it a little bit more carefully. So here are the different pathogens that they tested their biocontrol streptomyces against. So Streptomyces scabies is the potato scab pathogen. And then you can see Fusarium, Verticillium, Rhizoctonia, and another Fusarium. So what you're looking at here are Petri plates that have been, it's called the Hares assay. I won't go into it in huge detail, but basically you're testing the inhibitory activity in the soil. You take a little bit of soil from those plots, you dilute it, and then you plate it out across the Petri plate and let it grow. And so any bacteria showing up, they were looking in particular for Streptomyces bacteria from those soils. So we know how to influence so that the Streptomyces preferentially grow from the soil onto these plates. Then what you do is you lay another layer of agar over all those colonies that grew up, and then you put your pathogen down on top of that and spread it across the plate. And so the pathogen will start growing across that thin layer of agar. All the soil isolates are underneath. So if the soil isolates are producing any kind of antibiotic, it'll go into that thin layer of agar and start killing the pathogen. And so then you see these zones of inhibition. And so you can see across, especially like that top left plate, you can see that there were many isolates from the soil that once they laid down the pathogen across the surface, you could see they definitely have some antibiotic that was killing and leaving what we call a kill zone or an inhibition zone against the pathogen. And so the Fusarium, Verticillium, Rhizoctonia, and the other Fusarium, those are all fungi. And then the Streptomyces scabies is a bacteria. So all these pathogens you can see are getting inhibited by the Streptomyces bacteria that they isolated from these different soil systems in their test. And what they were finding is that the plant species is definitely influencing what's going on in terms of how well their associated Streptomyces were able to kill pathogens. Okay, so you can see these are, you don't need to know which plant did what, that's unimportant, but you can definitely see which plant's there, it does make a difference. You see different letters like that, that means they're statistically different from each other. And so, yeah, it matters. It matters what kind of plants are there in the ability of the Streptomyces that are there in the rhizosphere, their ability to inhibit those pathogens that we were looking at in the previous slide. Okay. Here they went back to the idea of if you just have a monoculture. So one means you have a monoculture of plants versus 16 different species in that plot. And again, you can see some really pretty data here where if you have a monoculture, you have a lot of microbes, a lot of Streptomyces, mean proportion of inhibitors. There's a lot of them that are capable of inhibiting those pathogens. On the other hand, you look down, on the far right where you have 16 different species. There's inhibition, but it's not nearly as good. So this is a little bit of a head scratcher because, we've been saying how, diversity of of ecosystems, generally a good thing, right? Like if you see a forest ecosystem, diversity of plants, generally a good thing. And yes, that's still true, but we're looking at a very specific idea here. We're looking at how do we make, in this case, the Streptomyces bacteria that hang out with these plants more inhibitory? How do we force them to be more inhibitory? That's what they need to do for biocontrol of these pathogens that attack crops. And so it looks like monoculture, right? Monoculture is the way to go. You get better inhibition if it's one species of plant, which kind of makes you think about, okay, well, there's a lot of organic farmers and permaculture farmers that are going away from a monoculture system. We know monoculture can be, there's several disadvantages to that. And yet in this particular case, it's showing an advantage in terms of if you have a monoculture, you're creating a way in which the Streptomyces bacteria, they're all fighting, essentially, they're doing a significant niche overlap. They're all fighting for the same kind of carbon source, the exudates, and it's making them very inhibitory. They're getting very hangry with each other. Okay. So if more nutrients are better, high glucose, high lignin, how come we see, we initially would think high diversity of plants would be a good thing because that gets you a lot of yummy carbohydrate coming out into the soil from the plants. So why? And so the more diverse plant communities have higher carbon, organic matter, and productivity, which in theory should give you nice inhibition. The bacteria should be able to inhibit. So why aren't they doing that? And so this data initially was a little surprising, but then when you start thinking about niche overlap, it starts to make more sense. And the idea of niche differentiation and kind of playing with that idea in order to force the microbe to do what you want it to do. And so you saw this already, where if there's significant niche overlap, so they're sharing the same nutrients, and at 100%, it's like, all the same. You get nice inhibition under that state. Here on the right, you're seeing the idea of niche differentiation showing up here. Here you have niche overlap on the top and niche differentiation on the bottom. So if you have a plot of plants with high diversity, all those 16 different species, basically what the hypothesis is that those bacteria are able to do niche differentiation. It's kind of like if at these tables, like we were all out there, right, waiting for the door to get unlocked. And what if, before I let you in, on each table, I put sugar cookies on every table. And then I let you in and said, go to whatever table, but it's all sugar cookies. It's all the same. And most of us like sugar cookies probably, right? You might fight over that a little bit. But if I have different cookies, chocolate chip, oatmeal, sugar, snickerdoodle, and on, you'll have preferences. I like snickerdoodle. I'll eat this. Oh, you can have the sugar cookie. Yeah, I like sugar cookie. And so you start to differentiate what you're going to eat, right? You have all these different lovely sugar cookies or different cookies. And so that's kind of like the niche differentiation. If you have a lot of different carbon sources in the soil, the microbes tend to not fight with each other. They kind of get their own niche and like, I'm happy with this carbon source. You're happy with that. It's all good. I'm not going to fight you because it's, if you're producing antibiotics, I don't know if you've ever seen an antibiotic physiological pathway. It's complicated. And it's an investment of resources if you're going to make that antibiotic. So if you don't have to do it, don't. And so if you have 16 different species of plants, all exuding a bunch of different chemicals that these bacteria can take and use, you can start to appreciate that you're going to get that differentiation occurring, and then they won't turn on their antibiotic production. But if you can force them to have niche overlap, it gets them fighting each other. And the way they do that is through antibiotic production. And so the monoculture idea is actually a better situation in this particular idea of how do we make these streptomyces more inhibitory toward these different pathogens. So this is a different way of showing that niche differentiation. Niche escape is more like niche differentiation and niche overlap. Does that make sense? Okay. So sometimes a monoculture is not all bad. It has some disadvantages in other aspects, but in this particular case, when they were trying to find those inhibitory properties, the phenotype, then actually in this flow chart, you want to force the top flow there. You don't want a lot of nutrient diversity. You want them fighting for the same carbon source, essentially. And if you do that, that's like an arms race amongst themselves. They'll kick in their antibiotic production and become highly inhibitory to the pathogens. So when they started this experiment, they were met with some interesting puzzling results. But once you started thinking about niche differentiation versus niche overlap, it started to fall better into place. And so they've done other studies since then and have backed this up. But summary plant host. So who is the plant and how many are there? Is it a monoculture? Is it a diverse mix? Definitely has an impact on what's going on in the rhizosphere. And in this case, looking at inhibitory profiles of those streptomyces. Nutrients, is it, diverse nutrients or low diversity in the nutrient source? That definitely has an impact. And so they're saying if you're going into the agricultural fields where a lot of fields are still monocultures, keeping this in mind, especially if you have a crop that is prone to, these certain pathogens. And so this idea of making the microbes fight is the way to get them to ramp up their antibiotic profiles, which is good for killing off the pathogen. Questions on that? Where did that go? So we've talked about niche partitioning, but I also wanted to introduce this idea of niche overlap, that you can tweak that, and under the right states, you can influence heavily how in this case, biocontrol agents act. Okay. All right. The next, I want to get into the next slide set a little bit before we break and get into talking about the group projects. It's going to be kind of a continuing on a little bit with this idea in some regards, because on the next topic, I want to get into kind of looking at what is going on in the agricultural realm. So I kind of call this the future of agriculture ideas that are out there right now. And this is, I wanted to highlight a few companies that are kind of front runners in some of their innovations on how to grow food more sustainably and do more yield, since we're going to have so many more mouths to feed and we only have so much land to put into growing food. So this is a company called Jord Bioscience. It's actually my postdoc advisor, Dr. Linda Kinkle. She started this company because she got tired of, she was a rock star at the University of Minnesota in terms of winning many $1 million worth of grant money, did a ton of research, very active lab, looking at a lot of it was biocontrol of plant diseases. And she was finding all kinds of cool microbes, but it wasn't getting out into the real world where farmers were using it, growers were using it. So she started Jord Bioscience to bridge that gap. And so her focus on this with this company is, I think she's got over 6,000 different microbes that she's collected from all over the world, many of them coming from agricultural systems, and has done a ton of research on how they interact with each other, how they influence each other, and how they could be beneficial for the different plants in crop production. And so here, um, in general, they're looking at, this one is focused on phosphate. Phosphate availability is very tough, as we know. Plants have a tough time with getting access to that. So part of their company, that's part of their focus, is to help the farmers get the right mix of microbes to improve phosphate solubilization. But that's not the only thing that they're doing. With over 6,000 microbes, there's a lot going on. But, the company was created to translate nearly 4 decades of research. And so she's been looking at all kinds of different crop systems. She did a lot of work in native prairie grass soils. Have you heard of LITER sites? They're called LTER is the acronym. Stands for long-term ecological research. And so there's native prairie systems at different LITER sites owned or run by different universities. And there's one in Minnesota somewhere, I'm blanking on the city now, where so she was studying prairie systems, undisturbed prairie systems and different plot constructions, so the previous study I just talked to you about, that was at a native prairie soil where they had different plots of soils, some were in monoculture and all the way up to 16 different species over long term. And so the idea there is that the microbes that hang out with those plants in this long-term ecosystem are awesome to study because it gives you so much information on their co-evolutionary partnerships and how they're influencing each other. And so she's done a lot of that work. So collected over, 6,500 microbial isolates and has studied those all the way from basic plate assays to doing machine learning on them to figure out what they can do, and then turning that into a way to help growers enhance crop production, drive more consistent results. And so this was an interview that she was doing where, again, she was just saying that she was finding all this great data in the silo of academia, but it wasn't getting out there in the real world. And so she says she hopes her experience will inspire other researchers to increase the impact of their academic work through commercialization activities and working with industry partners. And I think our world is getting more that way, but I wish it would speed up. We need more of this because a lot of great studies are going on at the universities, but then they often stumble when it comes time to get that out into real world application. Just want to impress on you, this is a lot of work over a long period of time. She started this in the 1990s at the University of Minnesota. And so we're talking a long stretch of time of really hard research, lots of grant funding through this, all millions of dollars put toward this, and finally getting into the point where she's starting to work with the growers more directly. And this is some information taken from the website. And so one of the nasty pathogens out there is called Pythium. It's a fungus. And so they were doing some case study work on that with some of the microbe cocktail mixtures that they have in order to try to control. And so her company is kind of in the infancy, young stages. She's still, it's a pretty new company, but they're having some really good results with the case studies that they've been doing. So often when these companies start up, then they partnership with universities to do some case study work. Like if I give you my product, will you put it into your field in an experimental design so that we can get some data? And so that's usually the way that they work it here. So there's some of the fungi growing on the plates. And so here, again, just saying if we go about this the right way, where we have the right combination of microbes or whatever, do you need more phosphate solubilization? Do you need biocontrol agents? What's going on here? And so she is working on that. And I just saw just on Halloween, she was awarded the 2025 Top Agri-Food Pioneers by the World Food Prize Foundation, which is a very, very high honor. So good on her that she was able to get that recognition. She's been going at this, like I said, for a long time. And so the Jord Bioscience company is relatively small, but it's obviously she's making some good progress on this. So I think we'll be hearing a lot more about that over time. This was an article that came out in, I think, around 2018, where they were just discussing like what's new. And this one is again, looking at the microbes that are hanging out with the crop plants. In this case, they're showing corn, but lots of different, it's not just corn. And so one of the big ideas out there that a lot of growers are looking at is how do we use these microbes in the soil? Now we know enough about them that we understand how important they are for the plant. How can we use that knowledge now to really maximize crop production, yield, nutrient density? And they know that, looking at the microbes is one huge way to do that. And it's very sustainable compared to other methods. Thought it was interesting that Bayer Incorporation, Bayer Aspirin, they're getting involved in this too. So there's several companies that have been coming online. So self-fertilizing crops is a big one. I wanted to talk to you a little bit about that. So this company called Pivot Bio has been established longer than Jord Bioscience. And so they're a little further along. But what they're really focusing in on is how do we increase nitrogen in the soil without, let's reduce our nitrogen input with the fertilizers that we put into the system. Let's get the microbes in the soil to do this. And I mean, we know already, you talked about nitrogen fixation, but that's not exactly what they're talking about, like the whole idea of a nodule formation and the microbes hanging out in the root tissue. They're talking more about free living nitrogen fixers. There's all kinds of microbes that just hang out in the rhizosphere that can fix nitrogen. They don't have to form a relationship with the plant and live in the nodules and all of that. They're just hanging out in the soil and they're fixing nitrogen. And so I think I showed you this. It's part of a table of there's some free living ones that just do this. And so the idea here is okay, we know there's all these nitrogen fixers in the soil. But they're planting their crop in fields that have been heavily fertilized over decades sometimes, right? All these artificial fertilizers. So if you're a microbe and I tell you that, fixing nitrogen can be costly. You're using resources to build all the necessary physiological steps, right? It's costly. So if you're a microbe hanging out in the soil and a bunch of nitrogen comes raining down on you in the soil, you'll probably go, I have all this lovely nitrogen coming at me. I'm not going to fix nitrogen because that's expensive for me. And so what they're saying is, we know that there's these microbes in the soil that can fix nitrogen. It's just they don't because they've been living in fields where nitrogen's been provided to them. They've been spoon fed. And so they've turned off those genes and said, no, I don't want to do it. I'm getting plenty of nitrogen the other way. And so they said, basically, we had to turn those genes back on. We had to force the situation. So they needed to apply a selective pressure on those microbes to say, hey, get those genes up and running, start fixing nitrogen. And then the other idea is, of course, give some of that to the plants so that the crop can grow with less synthetic fertilizers added into the system. And so that's what they've been working on. And, they say the crop microbiome holds the future for fertilizers. We should be able, if we can do this smart, we should be able to reduce inorganic fertilizer input by humans and instead just create conditions for the microbes that are in the soil that can do this anyways, make them do it. And so here, I would like you to think a little bit about what are, the downsides in terms of we need to do fertilization right now anyways. I mean, that's been a huge boon to crop production, definitely, but it has downsides. We've talked a little bit about that. You can get all the runoff, fertilizer runoff that starts to pollute water table, and rivers and streams and oceans. I think it even says here, worldwide fertilizer linked pollution is responsible for more than 500 dead zones. Places so toxic that nothing lives. One of the largest is where the Mississippi River empties into the Gulf of Mexico. It's nearly the size of New Jersey. Isn't that a lovely thought? So they're saying, yeah, we need nitrogen in the soil, but it's definitely creating issues, greenhouse gases, pollution into our water sources. So they're saying we have a renewable, clean alternative. So they didn't try to genetically alter the plant to become like a legume plant where it can hang out with these bacteria and form nodules, although they are working on stuff like that. Instead, they're saying, let's just take those free living microbes that hang out in the rhizosphere. Let's make them turn on their genes that have been dormant. And so they didn't genetically modify these organisms, so they don't fall under the category of GMOs. They just figured out a way to get those genes up and running and took a lot of work. So trillions of soil microbes that they mapped. And they, of course, wanted to find microbes that hang out consistently with the crop of interest. Usually, I think they started in corn. And we evaluate billions of ways to edit the microbe's own genetic code. So they're using a lot of machine learning and synthetic biology concepts on that. We do not introduce genes from other organisms. They just work within the microbe's own genome. So there's less controversy about this idea. And then what they do is they take the corn seed that they're going to plant in the field and they coat it with these microbes. So they coat the seed and then plant the seed. And as soon as the roots emerge, the microbes are there to start interacting with the roots. And so they start to hang out like either in the soil right next to the roots or right on the roots. Probably some of them are going endophytic to some extent. And they start to fix nitrogen. So this is one of the papers they published on it. So genetic remodeling. So they're not GMOs in the true sense. Diazotrophs, that word, soil diazotroph. Di means two, right? Azo, nitrogen. Troph means to use or to eat. So if someone says, oh yeah, I got those diazotrophs from the soil, they're just talking about microbes that can fix nitrogen. That's what they're talking about. And so they're saying we can partially reduce, synthetic nitrogen that we put into the system if we can get these microbes up and running. And their model plant was corn. So this next figure is taken from the paper. What I want you to mostly focus on is figure A up top. So they're saying business as usual. They're saying this is what is currently being done, where the microbes are quiet. They have the gene for nitrogen fixation, but they won't do it because they have a lot of input of nitrogen fertilizer coming into that field soil. And so with that higher amount, they figure I'm not going to do it myself because I have plenty of nitrogen around me. I'll just use that. But of course, then you get all the negative inputs, the greenhouse gas, the runoff and pollution. Then they say, let's compare it to what we have been able to do now with gene editing. And so now they're saying you can get the microbes. Not only have they created the way to turn those genes on, so the microbes turn the genes on, but they also made it so that the microbes excrete out at a higher rate the nitrogen, the fixed nitrogen, to the plant. And so they're not keeping it all to themselves, they're excreting it out at higher rates than typical. So they really thought that through. And because of that, they say we should be able to use lower nitrogen amounts, which should help with more sustainable practices. The stuff on the bottom here is just some of the details of how they did it, which we don't need to get into. But you can see it's complicated. It took them a long time. This is a lot of effort by a lot of different scientists. Okay, let's stop there. Let's take a break for about 10 minutes. So a little like 3:01-ish. We'll come back and think a little bit more on this and talk about our projects, group projects. Hmm. So I will get that back to you soon. But in the meantime, you can get going on this next assignment. This is a table or group. So you can group up if you want to join theirs too. And I know there's some people missing today, but the idea here is it looks like we'll have 4 tables of students. And then the idea is to agree on a topic, a paper. I'd like it to be a primary research paper where they, show explain methods and results and conclusions rather than a review paper. I need to know what it is because I have to read it as well so that when we're in our sessions together, I can help facilitate what's going on. But I think it would be anything's kind of fair game, something of interest. It doesn't have to be something we're currently covering. Remember back on the very first week where I said, what do you want to learn? What do you want to get from this class? And some of you mentioned some things that I haven't had a chance to talk about yet. So if it's something like that you want to pull in, great. If it's something we've already covered, but just not me directly covering it, fine. So I want it to be more of a consensus at your table of a cool paper that you'd like to talk about. Then the idea is to do basically what you did for that first assignment, but as a group. And then we'll take turns presenting. And the way I've done this in the past, it seems to work, is I will upload it, or you'll upload it into EduCat. I'll get access to it and I'll have it on my laptop and you'll just sit at your tables. And if it's your turn, you just chat about it and you take turns talking about it. So not just one person at each table. Everyone will have some input on different aspects of the presentation. You've probably done this maybe at some other classes, but I think this would be a good way to pick some things that you're interested in and that I might not have time to cover in a more formal setting. So that's the general plan. I don't know on the first due date. I have to look. I can't believe there's only 4 more weeks of the semester. I don't know what the heck happened. So yeah, I mean, we have break in there too, but it's like, wait a minute. So that means we have 8 more sessions together. So we got to fit all this in. Plus, I want to still cover phytoremediation. So I'm covering kind of like the future of ag, and then phytoremediation will be my last topic. Um, so keep that in mind too. So yeah, we got to fit this all in. So I want at least 2 papers, 2 group papers done and so usually what I've done in the past is you present on one of them and the other one probably not. And so we kind of take turns, but each table will do prepare 2 presentations. Let's put it that way. Am I making sense? It's Monday. All right. So what I'd like you to do is sit while you're here together face to face, and think about what kind of paper. Are you fairly familiar with Google Scholar for searching? Do you know how to do the cite? Like if you find a paper. Oh, a few more things I have to say. If you find a paper, do you know you can click on that cited by below the paper? Have you done that before? And then it'll pull up all the papers that have cited that paper. So like if you find a paper, the reason I'm saying this is I would like you to find a more recent paper. So if you find a paper, but it's got an older date, if you click on the cited by, you might find another paper that's on the same topic that's newer. So I'd like you to find something that's ideally within the years 2018 to 2025 in that time span. Unless there's a really, really, really cool paper that you're just like, but I really want this one. I can give you some feedback. There might be some wiggle room on that. All right. So why don't you get in your groups and see if you can come to a consensus on what your first paper will be.